Namaste, and welcome back to BioFod. We are continuing our exploration of human physiology, now focusing on the content as presented in the new 2024-25 NCERT textbook. Today, we are diving into Chapter 14, Breathing and Exchange of Gases. This is a fundamental process that keeps us alive, and we are going to explore how it works in detail. Understanding the breathing and exchange of gases is crucial for understanding human physiology and health. So, grab your new NCERT textbooks, maybe take a deep breath, and get ready to explore the amazing world of respiration right here on BioFod. Let's breathe in some knowledge. As you have read earlier, oxygen, O2, is utilized by the organisms to indirectly break down simple molecules like glucose, amino acids, fatty acids, etc., to derive energy to perform various activities. Carbon dioxide, CO2, which is harmful, is also released during the above catabolic reactions. It is, therefore, evident that O2 has to be continuously provided to the cells, and CO2 produced by the cells have to be released out. This process of exchange of O2 from the atmosphere with CO2 produced by the cells is called breathing, commonly known as respiration. Place your hands on your chest. You can feel the chest moving up and down. You know that it is due to breathing. How do we breathe? The respiratory organs and the mechanism of breathing are described in the following sections of this chapter. 14.1 Respiratory organs. Mechanisms of breathing vary among different groups of animals, depending mainly on their habitats and levels of organization. Lower invertebrates like sponges, coelenterate, flatworms, etc. exchange O2 with CO2 by simple diffusion over their entire body surface. Earthworms use their moist cuticle, and insects have a network of tubes, tracheal tubes, to transport atmospheric air within the body. Special vascularized structures called gills, branchial respiration, are used by most of the aquatic arthropods and mollusks, whereas vascularized bags called lungs, pulmonary respiration, are used by the terrestrial forms for the exchange of gases. Among vertebrates, fishes use gills, whereas amphibians, reptiles, birds and mammals respire through lungs. Amphibians like frogs can respire through their moist skin, cutaneous respiration also. 4.1.1 Human respiratory system. We have a pair of external nostrils opening out above the upper lips. It leads to a nasal chamber through the nasal passage. The nasal chamber opens into the pharynx, a portion of which is the common passage for food and air. The pharynx opens through the larynx region into the trachea. Larynx is a cartilaginous box which helps in sound production and hence called the sound box. During swallowing, glottis can be covered by a thin elastic cartilaginous flap called epiglottis to prevent the entry of food into the larynx. Trachea is a straight tube extending up to the mid-thoracic cavity, which divides at the level of fifth thoracic vertebra into a right and left primary bronchi. Each bronchi undergoes repeated divisions to form the secondary and tertiary bronchi, and bronchioles ending up in very thin terminal bronchioles. The trachea, primary, secondary and tertiary bronchi, and initial bronchioles are supported by incomplete cartilaginous rings. Each terminal bronchiole gives rise to a number of very thin, irregular walled and vascularized bag-like structures called alveoli. The branching Network of bronchi, bronchioles and alveoli comprise the lungs. Figure 14.1. We have two lungs, which are covered by a double-layered pleura, with pleural fluid between them. It reduces friction on the lung surface. The outer pleural membrane is in close contact with the thoracic lining, whereas the inner pleural membrane is in contact with the lung surface. The part starting with the external nostrils up to the terminal bronchioles constitute the conducting part, whereas the alveoli and their ducts form the respiratory or exchange part of the respiratory system. The conducting part transports the atmospheric air to the alveoli, clears it from foreign particles, humidifies, and also brings the air to body temperature. Exchange part is the site of actual diffusion of O2 and CO2 between blood and atmospheric air. The lungs are situated in the thoracic chamber, which is anatomically an airtight chamber. The thoracic chamber is formed dorsally by the vertebral column, ventrally by the sternum, laterally by the ribs, and on the lower side by the dome-shaped diaphragm. The anatomical setup of lungs in thorax is such that any change in the volume of the thoracic cavity will be reflected in the lung pulmonary cavity. Such an arrangement is essential for breathing, as we cannot directly alter the pulmonary volume. Respiration involves the following steps. I. Breathing or pulmonary ventilation, by which atmospheric air is drawn in and CO2 rich alveolar air is released out. 2. 3. Diffusion of gases, O2 and CO2 across alveolar membrane. Transport of gases by the blood. 4. Diffusion of O2 and CO2 between blood and tissues. Utilization of O2 by the cells for catabolic reactions and resultant release of CO2. Cellular respiration as dealt in the chapter 12. 14.2 Mechanism of breathing. Breathing involves two stages. Inspiration, during which atmospheric air is drawn in, and expiration, by which the alveolar air is released out. The movement of air into and out of the lungs is carried out by creating a pressure gradient between the lungs and the atmosphere. Inspiration can occur if the pressure within the lungs, intrapulmonary pressure, is less than the atmospheric pressure, i.e., there is a negative pressure in the lungs with respect to atmospheric pressure. Similarly, expiration takes place when the intrapulmonary pressure is higher than the atmospheric pressure. The diaphragm and a specialized set of muscles, external and internal intercostals between the ribs, help in generation of such gradients. Inspiration is initiated by the contraction of diaphragm, which increases the volume of thoracic chamber in the anteroposterior axis. The contraction of external intercostal muscles lifts up the ribs and the sternum, causing an increase in the volume of the thoracic chamber in the dorsoventral axis. The overall increase in the thoracic volume causes a similar increase in pulmonary volume. An increase in pulmonary volume decreases the intrapulmonary pressure to less than the atmospheric pressure, which forces the air from outside to move into the lungs. I. Inspiration, figure 14.2a. Relaxation of the diaphragm and the intercostal muscles returns the diaphragm and sternum to their normal positions and reduce the thoracic volume and thereby the pulmonary volume. This leads to an increase in intrapulmonary pressure to slightly above the atmospheric pressure, causing the expulsion of air from the lungs, i.e., expiration, figure 14.2b. We have The ability to increase the strength of inspiration and expiration with the help of additional muscles in the abdomen. On an average, a healthy human breathes 12 to 16 times minute. The volume of air involved in breathing movements can be estimated by using a spirometer, which helps in clinical assessment of pulmonary functions. 4.2.1 Respiratory volumes and capacities. Tidal volume, TV volume of air inspired or expired during a normal respiration. It is approx 500 ml, i.e., a healthy man can inspire or expire approximately 6,000 to 8,000 ml of air per minute. Inspiratory reserve volume, IRV. Additional volume of air a person can inspire by a forcible inspiration. This averages 2,500 ml to 3,000 ml. Expiratory reserve volume, ERV. Additional volume of air a person can expire by a forcible expiration. This averages 1,000 ml to 1,100 ml. Residual volume, RV, volume of air remaining in the lungs even after a forcible expiration. This averages 1,100 ml to 1,200 ml. By adding up a few respiratory volumes described above, one can derive various pulmonary capacities, which can be used in clinical diagnosis. Inspiratory capacity, total volume of air a person can inspire after a normal expiration. This includes tidal volume and inspiratory reserve volume, TV plus IRV. Expiratory capacity, EC, total volume of air a person can expire after a normal inspiration. This includes tidal volume and expiratory reserve volume, TV plus ERV. Functional residual capacity, FRC, volume of air that will remain in the lungs after a normal expiration. This includes ERV plus RV. Vital capacity, VC, the maximum volume of air a person can breathe in after a forced expiration. This includes ERV, TV and IRV, or the maximum volume of air a person can breathe out after a forced inspiration. Total lung capacity, TLC, total volume of air accommodated in the lungs at the end of a forced inspiration. This includes RV, ERV, TV and IRV, or vital capacity plus residual volume. 14.3 Exchange of gases. Alveoli are the primary sites of exchange of gases. Exchange of gases also occur between blood and tissues. O2 and CO2 are exchanged in these sites by simple diffusion, mainly based on pressure concentration gradient, solubility of the gases, as well as the thickness of the membranes. Involved in diffusion are also some important factors that can affect the rate of diffusion. Pressure contributed by an individual gas in a mixture of gases is called partial pressure and is represented as PO2 for oxygen and PCO2 for carbon dioxide. Partial pressures of these two gases in the atmospheric air and the two sites of diffusion are given in Table 14.1 and in Figure 14.3. The data given in the table clearly indicates a concentration gradient for oxygen from alveoli to blood and blood to tissues. Similarly, a gradient is present for CO2 in the opposite direction, i.e., from tissues to blood and blood to alveoli. As the solubility of CO2 is 20 to 25 times higher than that of O2, the amount of CO2 that can diffuse through the diffusion Membrane per unit difference in partial pressure is much higher compared to that of O2. The diffusion membrane is made up of three major layers. Figure 14.4, namely, the thin squamous epithelium of alveoli, the endothelium of alveolar capillaries and the basement substance composed of a thin basement membrane supporting the squamous epithelium and the basement membrane surrounding the single-layer endothelial cells of capillaries in between them. However, its total thickness is much less than a millimeter. Therefore, all the factors in our body are favorable for diffusion of O2 from alveoli to tissues and that of CO2 from tissues to alveoli. 14.4 Transport of gases. Blood is the medium of transport for O2 and CO2. About 97% of O2 is transported by RBCs in the blood. The remaining 3% of O2 is carried in a dissolved state through the plasma. Nearly 20 to 25% of CO2 is transported by RBCs, whereas 70% of it is carried as bicarbonate. About 7% of CO2 is carried in a dissolved state through plasma. 14.4.1 Transport of oxygen. Hemoglobin is a red-colored iron-containing pigment present in the RBCs. O2 can bind with hemoglobin in a reversible manner to form oxyhemoglobin. Each hemoglobin molecule can carry a maximum of 4 molecules of O2. Binding of oxygen with hemoglobin is primarily related to partial pressure of O2, partial pressure of CO2, hydrogen ion concentration and temperature are the other factors which can interfere with this binding. A sigmoid curve is obtained when percentage saturation of hemoglobin with O2 is plotted against the PO2. This curve is called the oxygen dissociation curve, figure 14.5, and is highly useful in studying the effect of factors like PCO2, H+ concentration, etc. on binding of O2 with hemoglobin. In the alveoli, where there is high PO2, low PCO2, lesser H+ concentration, and lower temperature, the factors are all favorable for the formation of oxyhemoglobin, whereas in the tissues, where low PO2, high PCO2, high H+ concentration and higher temperature exist, the conditions are favorable for dissociation of oxygen from the oxyhemoglobin. This clearly indicates that O2 gets bound to hemoglobin in the lung surface and gets dissociated at the tissues. Every 100 milliliters of oxygenated blood can deliver around 5 milliliters of O2 to the tissues under normal physiological conditions. 14.4.2 Transport of carbon dioxide. CO2 is carried by hemoglobin as carbaminohemoglobin, about 20 to 25%. This binding is related to the partial pressure of CO2. PO2 is a major factor which could affect this binding. When PCO2 is high and PO2 is low, as in the tissues, more binding of carbon dioxide occurs. Whereas, when the PCO2 is low and PO2 is high, as in the alveoli, dissociation of CO2 from carbaminohemoglobin takes place, i.e., CO2, which is bound to hemoglobin from the tissues, is delivered at the alveoli. RBCs contain A very high concentration of the enzyme, carbonic anhydrase and minute quantities of the same is present in the plasma too. This enzyme facilitates the following reaction in both directions. CO2 plus H2O in presence of carbonic anhydrase gives H2CO3 and then again in presence of carbonic anhydrase gives HCO3 plus H plus. At the tissue site where partial pressure of CO2 is high due to catabolism, CO2 diffuses into blood, RBCs and plasma and forms HCO3 and H plus, at the alveolar site where PCO2 is low, the reaction proceeds in the opposite direction leading to the formation of CO2 and H2O. Thus, CO2 trapped as bicarbonate at the tissue level and transported to the alveoli is released out as CO2. Figure 14.4. Every 100 milliliters of deoxygenated blood delivers approximately 4 milliliters of CO2 to the alveoli. 14.5 Regulation of respiration. Human beings have a significant ability to maintain and moderate the respiratory rhythm to suit the demands of the body tissues. This is done by the neural system, a specialized center present in the medulla region of the brain called Respiratory Rhythm Center is primarily responsible for this regulation. Another center present in the pons region of the brain, called Pneumotaxic Center, can moderate the functions of the respiratory rhythm center. Neural signal from this center can reduce the duration of inspiration and thereby alter the respiratory rate. A chemosensitive area is situated adjacent to the rhythm center, which is highly sensitive to CO2 and hydrogen ions. Increase in these substances can activate this center, which in turn can signal the rhythm center to make necessary adjustments in the respiratory process by which these substances can be eliminated. Receptors associated with aortic arch and carotid artery also can recognize changes in CO2 and H plus concentration and send necessary signals to the rhythm center for remedial actions. The role of oxygen in the regulation of respiratory rhythm is quite insignificant. 14.6 Disorders of respiratory system. Asthma is a difficulty in breathing causing wheezing due to inflammation of bronchi and bronchioles. Emphysema is a chronic disorder in which alveolar walls are damaged, due to which respiratory surface is decreased. One of the major causes of this is cigarette smoking. Occupational respiratory disorders in certain industries, especially those involving grinding or stone breaking, so much dust is produced that the defense mechanism of the body cannot fully cope with the situation. Long exposure can give rise to inflammation leading to fibrosis, proliferation of fibrous tissues, and thus causing serious lung damage. Workers in such industries should wear protective masks. That was the chapter 14 according to new NCERT. Let's recap what we have learnt through. Summary. Cells utilize oxygen for metabolism and produce energy along with substances like carbon dioxide, which is harmful. Animals have evolved different mechanisms for the transport of oxygen to the cells and for the removal of carbon dioxide from there. We have a well-developed respiratory system comprising two lungs and associated air passages to perform this function. The first step in respiration is breathing, by which atmospheric air is taken in, inspiration, and the alveolar air is released out, expiration. Exchange of O2 and CO2 between deoxygenated blood and alveoli transport of these gases throughout the body by blood, exchange of O2 and CO2 between the oxygenated blood and tissues and utilization of O2 by the cells, cellular respiration, are the other steps involved. Inspiration and expiration are carried out by creating pressure gradients between the atmosphere and the alveoli with the help of specialized muscles, intercostals and diaphragm. Volumes of air involved in these activities can be estimated with the help of spirometer and are of clinical significance. Exchange of O2 and CO2 at the alveoli and tissues occur by diffusion. Rate of diffusion is dependent on the partial pressure gradients of O2, PO2 and CO2, PCO2, their solubility as well as the thickness of the diffusion surface. These factors in our body facilitate diffusion of O2 from the alveoli to the deoxygenated blood as well as from the oxygenated blood to the tissues. The factors are favorable for the diffusion of CO2 in the opposite direction, i.e., from tissues to alveoli. Oxygen is transported mainly as oxyhemoglobin in the alveoli where PO2 is higher. O2 gets bound to hemoglobin, which is easily dissociated at the tissues where PO2 is low and PCO2 and H plus concentration are high. Nearly 70% of carbon dioxide is transported as bicarbonate, HCO3, with the help of the enzyme carbonic anhydrase. 20 to 25% of carbon dioxide is carried by hemoglobin as carbaminohemoglobin. In the tissues where PCO2 is high, it gets bound to blood, whereas in the alveoli where PCO2 is low and PO2 is high, it gets removed from the blood. Respiratory rhythm is maintained by the respiratory center in the medulla region of brain, a pneumotaxic center in the pons region of the brain, and a chemosensitive area in the medulla can alter respiratory mechanism. And that brings us to the end of episode 17 of BioFod, our exploration of breathing and exchange of gases. I hope you now have a clearer understanding of how your respiratory system works. As always, the description box is packed with helpful resources, including diagrams of the respiratory system, animations of gas exchange, links to health information, or previous episodes. I encourage you to explore these resources further to solidify your understanding. And please share your thoughts. We love hearing from our listeners. Leave a comment or review, and let us know what you found most interesting about this episode. In our next episode, we'll continue our journey through human physiology with Chapter 15, Body Fluids and Circulation. We'll explore how our bodies transport essential substances throughout the body. So, be sure to subscribe to UD Lectures so you don't miss the next step in our exploration of the human body. Thank you for joining me on BioFod, the podcast series brought to you by UD Lectures. Until next time, keep your knowledge circulating.